So to the next speaker, it's going to be Emerson Rojas from the Texas Tech University Health Science Center in the United States. So Emerson, please put your presentation on. I'm going to stop sharing mine.
Okay, can you hear me?
Yeah, please activate your camera as well.
Okay,
I think.
I cannot. You can start.
I just did it.
Okay, yeah.
Yeah, it's there.
It should work, right? Okay. So let me this one here. Can you see? Can you hear me well?
Yeah, please adjust your camera because we cannot see your head.
Oh, sorry about that. Let me see. Oh yeah. Okay. Yeah, that's better. So, well, first of all, I want to thank the organizers for the opportunity to show part of my work here. Today, I'm going to tell you a story about the last part of my PhD thesis that I did here in Valparaiso, Chile. As you can see, this was an amazing place to work, not only because these great views that we have every day, but also because I had the pleasure to work with amazing people. These are amazing scientists and biophysicists, and I wanted to thank them first for all the fun that we had together, like doing science. So during my PhD, I worked with this channel that is the voltage-gated proton channel, or Hv1. And this one is expressed in a different variety of tissues, as you can see in red in the figure. We don't know actually the physiological role of many of them. However, we do know that it has function in the immune cells. Actually, the channel is expressed in both the myeloid and the lymphoid cells, and it has an essential role producing the immune response, which is highly relevant, especially in these times that we are living, right? So in these cells, Hv1 is functionally coupled to the ΔpH oxidase complex, or NOx. This complex produces superoxide by oxidation of ΔpH, and during this reaction, protons are produced in the intracellular part, as you can see, and Hv1 dissipates this pH gradient that forms to sustain the activity of ΔpH oxidase and the production of reactive oxygen species. So to accomplish that function, Hv1 has unique structure and biophysical properties as well. So we do know that the channel form dimers, as you can see in this cartoon. The opening of the channel is voltage dependent, also is pH dependent. Each subunit has its own permeation pathway that is highly selective to protons, and they open in a cooperative manner. So actually, we use a simpler model to study Hv1 that is produced when you delete the N and C terminal domain. And that results in monomeric channels that maintain the biophysical properties of the dimer in absence of cooperativity. So from all these properties, maybe the most interesting one is its pH dependence, since the open probability of the channel does not depend on the internal and external pH value, but on the delta pH established across the membrane. So if we plot the conductance as a function of voltage, or GB curve, you can see that at higher delta pH values, this curve shifts to the left, which means that we need less electrical energy to open the channel at higher delta pHs. So we wanted to understand this property, and we wanted to use this simpler model to do that. And although we know that the channel, we knew that the channel is pH dependent, it was unknown if the delta pH dependent gating is still maintained in this channel. So to study this, we express the channel in Xenopus-Levy oocytes and measure the currents produced in inside-out membrane patches by voltage clamp. And we use recording solutions that doesn't have sodium, potassium, or calcium to avoid any contamination with native currents of the oocyte. So here you can see a record at ΔpH 0. We avoid proton depletions or accumulations using high concentration of buffers, measuring the reversal potential after each measurement, and using this protocol, this voltage protocol that consists in shorter depolarizations at higher voltages. So taking that precautions, we measure the currents at different pHs. You can see here representative recorders also at ΔpH1 and at ΔpH2. And from the tail currents of these recordings, we can obtain the GV curve for different pHs. So when we plot that, you can see that at ΔpH 0, the three GV curves at the same ΔpH are located in similar positions in the voltage axis. And when we increase the ΔpH to 1, the GV curve shifted to the left. And an additional shift occurred when we increase further the ΔpH to 2. So that recapitulates the property that we saw in the dimer, which validates the monomeric channel to study the ΔpH dependent gating of Hv1. So knowing that, now we can start to do questions about the mechanics behind this property. So we actually previously have reported that the monomeric channel, in the monomeric channel, the activation of the voltage sensor occurs or precedes the opening of the permeation pathway. So we wanted to know, actually, we did a very simple question. That is, well, this ΔpH dependent gating is produced by modulation of the voltage sensor activation or the opening of the permeation pathway. So the GV curves that I showed actually cannot answer that question because proton currents are produced by opening of the permeation pathway in this case. So in order to study this other process, that the activation of the voltage sensor, we need to also measure gating currents. These currents are produced by charges on the voltage sensor that moves during activation of this structural domain. So we have first this transient current, this outward transient current or on-gating current that is produced when the voltage sensor activates. And then we have the off-gating current that is produced when the voltage sensor deactivates, moving in each case these charges that in this case are shown in the cartoon as positive charges. So from this record, we can obtain the charge displaced at each voltage, integrating the current as a function of time. And with that, we can plot the QV curve that actually contains the information that we are looking for. However, we have a problem because the gating currents are produced by the movement of few charges. So actually the currents are very small and they are hidden then by the higher magnitude of proton currents. So to record gating currents, we need to decrease proton currents or eliminate them somehow. So to solve that issue, we look for non-conducting mutants. It has been previously reported that mutations in the selectivity filter of the channel, that this is aspartate in S1, producing non-conducting channels. Here you see an example of human Hv1 mutant D112B, and it was not possible to record detectable proton currents even at a ΔpH 1.5. So this absence of current is not because a defective voltage sensor. That was demonstrated if you label channels in S4 with a fluorophore, you still can obtain, as you can see here, voltage-dependent fluorescence changes that indicates that the voltage sensor is still performing conformational changes. So this mutation seems ideal to do the studies that we want. So we introduced that mutation in our monomeric channel, and indeed we record robust gating currents of Hv1, as you can see in this recording. These currents were characterized by a fast component at the beginning of the on, followed by a very slow one. And similarly, in the off-gating current, you can see a very fast component at the beginning, followed by a slower one. So we always observe, as you can see in this magnification, a remnant of current at the end of the on gating current, especially at higher voltages. But despite that, we found that this mutant was the tool that we need to study the pH dependence of the voltage sensor activation. So we record gating currents at different pHs. You can see here a representative current at ΔpH 0, ΔpH 1, and ΔpH 2. And easily you can conclude that actually the pH is modulating the movement of the voltage sensor as the kinetics of decay of the gating currents change at different pHs. You can know that by the different time scales of this recording, for instance. However, we were interested more in how ΔpH changed the equilibrium. So to do that, we determine the QV curves at different pHs. And here you can see the QV curve at 3 different ΔpH, at ΔpH 0 and 3 different internal and external pHs, and they superimpose nicely, right? So when we increase the ΔpH to 1, actually QV curve shifted to the left, and an additional shift occurred at ΔpH 2. So this trend is the same, is similar that we observe in the GV curves of the conducting channel, demonstrating that the delta pH dependent gating is caused or is produced by modulation of the voltage sensor activation. So if we model the voltage sensor activation as a two-state model, we can represent this process by this energy profile at delta pH 0. And when we increase the delta pH this energy difference between the resting and active state of the voltage sensor decreases, which will produce the shifts that we observe in the QV and GV curves. So both, in this case, both voltage and delta pH are able to decrease this energy difference. However, how much energy stored in the delta pH is used to activate the voltage sensor? To answer that, we can express the free energy of the resting state as a function of the proton chemical potential, the internal proton chemical potential, and the internal electrical potential. And similarly, express the free energy of the active state as a function of the external potentials. And with that, we can obtain this free energy difference, which is function of delta pH and the transmembrane voltage. And you can see that these equations say that at higher delta pH and higher voltages, this free energy is going to decrease with this according with the data that we obtain, right? So we use this expression of the free energy to insert it in the QV equation, and with that we can globally fit all our data to obtain this parameter epsilon. This parameter epsilon is quantifying how much energy stored in the delta pH is used to activate the voltage sensor. So we obtain a value of 0.57, which means that around 60% of this energy in the proton chemical potential is used to activate the Hv1 voltage sensor. So in summary, we studied the delta pH dependent gating in Hv1 using the monomeric channel, and measuring gating currents of Hv1, we could conclude that the delta pH dependent gating is produced by the modulation of the voltage sensor activation. And around 60% of the energy stored is used to activate the voltage sensor of Hv1. However, how is the molecular mechanism that explains this property? Actually, we don't know, but I am currently working on that. I am now a PU postdoctoral fellow in Luis Cuello's lab, and here we are now studying the structure, function, and dynamics of purified human Hv1. And until now, we have managed to obtain pure protein, as you can see in the protein gel, and this protein is a good, it behaves as a good biochemical quality, as assessed by size exclusion chromatography. So I will have new exciting result to share with you in the future. So with that, I want to thank you for your attention, and I will be happy to answer any question that you have.
Emerson, thank you for a nice presentation and for keeping with the time. So we have one question here, and the question and answer from Gerardo de Palma. Is there a chance that the gating current is being modified or even generated when the mutation is performed? I ask this because the mutation involves an acidic residue that may carry an electric charge.
Okay, yeah, that is an interesting question, and we have studied the gating currents of Hv1 using another mutation that produce low conducting mutants. It's a low conducting mutant, so it just decreased the proton current, but not enough to like the mutant that I show you in this slide. So with that, we studied previously the gating currents. We studied the properties of it. We separate mathematically the proton currents from the gating currents. And then when we generate this other mutant, the first thing that we did was validate that, comparing that gating currents in the low conducting mutant with the gating currents in this one. That is a mutant that conducts even less protons. So they maintain the same properties. So we are sure that it's a good representation of the gating currents of Hv1. But it's true that we will like to the best way will be measuring the gating currents in the conducting channels, right? However, because of you can't eliminate protons of the solutions, or there is no still any kind of blocker that separate the permeation from the activation of the voltage sensor, we couldn't do that in that way. So we have to use these other approximations to make some conclusion about the process. Yeah, okay. It's a good point. Yeah.
Thank you. Now there's a question from Daniel Pelufo, please. Can you make your question, Daniel?
Yes. First, I like to say hello to my dear friend Rosangela and Antonio and all the Brazilian friends, and to remind Eduardo Peroso, he owes me a visit to Salto from long time ago. And so the question that I have for you is, in your gating currents from the non-conducting mutant, do you have an explanation for that non-zero remaining current? So this might be related to the previous question, but if you have gating currents only in a non-conducting mutant, the curve should go back to zero.
Yeah, sure. Yeah, that is absolutely true. And yeah, we have to find a methodology to assure that the charge during the on and the off were the same, and eliminate somehow. We did that with mathematical approach with fitting to assure that they were truly gating currents. However, I think we couldn't analyze that current because it was too small. However, just recent results that are unpublished suggest that current actually could be current from the channel. I don't know if proton currents or is not selective, but when you add GBI, that it's a blocker of the channel, an inhibitor actually, that remain current decreases and go to zero. So actually the channel is non-conducting. It keeps a small amount of current. However, even with that, we could analyze and the gating currents that we couldn't with the other low conducting that the proton current was higher, right? So yeah. Thank you for the question. Yeah.
Thank you.